Okay, welcome back from the coffee break. It is such a pleasure for me to introduce our first speaker of the second session. So we're delighted to have Dr. Ann Moscona here with us. She's the Sherry L. Morrison Professor at Columbia University in the departments of microbiology and immunology and of physiology and cellular biophysics. And your ears may have perked up at the Sherry Morrison chair because today we also have Sherry Morrison here as our distinguished colleague at the symposium. She might be still out at the coffee break. Oh no, she's right there. Okay, so welcome, Sherry. Anne was just installed in this chair in April, I understand, so congratulations. Anne is also a professor in the Department of Pediatrics at Columbia University Medical Center and is a practicing clinician there. And previously, she'd been on the faculty of the Mount Sinai Medical Center in New York and of the Cornell University Medical Center. Dr. Moscona's research is broad and interdisciplinary, and you might have gathered that from all the departments she's affiliated with, but is primarily focused on the paramyxoviruses. She works on common pediatric viruses, in particular parainfluenza, measles, and respiratory syncytial virus, but also on emerging lethal viruses like the Nipah and the Hendra. And I was asking her, is the Nipah the one in that movie? Did anyone see Contagion? Okay, Contagion, it's a great film. Has Anne's lab in it. So we need to hear more about that. Her lab is best known, though, not necessarily for being in that movie, but for elucidating the roles played by viral receptor binding proteins in activating the viral fusion process that facilitates the entry of the virus into its host cell. And she'll be speaking today on early steps of infection by paramyxoviruses. Thanks, Anne.
Thank you so much. Is the mic on? Yes? It's really an honor for me to be here and a pleasure to be celebrating Bill Gelbart and the award of the Seaborg Medal. And I'll be talking to you about early steps in infection by paramyxoviruses, a group of viruses I'll explain in a moment. But first of all, I have never worked with Bill. Bill and I met in a cafe in New York 16 years ago. This is a different cafe than the one that Avi was talking about. So this does, but this does, yes, this isn't the one you mentioned before, but this does seem to be a theme. So 16 years later, we're still trying to work together, and I'm hopeful that this event will be the start of that. So paramyxoviruses as a group take a major toll on child health, and if you look at the causes of death in children under 5, once you get through the neonatal period, most children are dying of respiratory infections. And when you take these respiratory infections, which are largely caused by paramyxoviruses, together with measles, which is another paramyxovirus, you really have the largest cause of death in kids under 5 after the newborn period. And these respiratory illnesses are mostly caused by parainfluenza virus, respiratory syncytial virus, and metapneumovirus, which are all paramyxoviruses, as well as influenza. A general schematic of the life cycle of paramyxoviruses, and then I'm going to focus in on the steps of entry. The paramyxoviruses are all enveloped viruses. The envelope consists of a membrane derived from the host cell during exit, during budding. And they are negative sense RNA viruses, which means that they enter a cell with their own RNA-driven RNA polymerase so that they can replicate their genome. And transcription and replication of the genome and transcription all takes place in the cytoplasm of the infected cell. Entry is initiated by binding of a receptor binding protein to a receptor, which in the case of parainfluenza that I'll focus on is a sialic acid containing receptor. And then fusion of the virus mediated by a different viral fusion protein into the cell and release of the contents into the cytoplasm. And then exit takes place by budding a virus through an area of the membrane into which the viral glycoproteins have been inserted. And then pinching off of the virus is accomplished by the neuraminidase, which releases a virus from being attached to the cell that it's budding out from. To focus in detail on the steps of entry, the first thing that occurs is binding of the receptor binding protein to target cell receptor, and then followed by activation of the fusion protein, a separate protein. In the work that's sort of a prelude to the work that I'm going to show you, we showed that actually, as opposed to what had been previously thought, the binding of the receptor binding protein to receptor, then the receptor binding protein then plays an active role in activating the fusion protein, which cannot function by itself in mediating entry. The fusion protein then undergoes a series of conformational changes to extend into the target cell, and then by a refolding step, which I'll show you more about, mediates fusion of the viral envelope with the target cell envelope and release of the contents into the cytoplasm. For parainfluenza, which is the virus that I'm going to focus on, the receptor binding protein is called the hemagglutinin neuraminidase, or HN, and the fusion protein is called F, or fusion. And again, the HN then actively triggers the fusion protein to extend, insert into the target, and the fusion protein then mediates the actual membrane fusion. To focus in on the steps in entry in a bit more detail, prior to the start of this process, in order for the virus to be infectious, F has to be cleaved from a proprotein, or F0, into its two active subunits. And during that cleavage, a fusion peptide is exposed at the N-terminus, which is then going to be what inserts into the target. So the first thing that occurs is that HN, the receptor binding protein, interacts with a sialic acid containing receptor on the target cell. Now, F has to already be in its cleaved and sort of ready state, but fusion readiness actually requires then a second activation step that occurs upon HN engagement with target receptor. HN actually triggers F to extend, insert its fusion peptide into the target membrane, and then refold in such a way that the heptad repeats at the N-terminus fold to meet the heptad repeats at the C-terminus, ultimately reaching the final conformational state of F, which is the most energetically stable, and that is what drives the fusion of the creation of a fusion pore and the merger of the viral and cell membranes. So I'm going to really focus in on a major interest of ours, which is how F is triggered by HN. What happens when HN binds receptor to actively trigger the onset of the fusion process? Before doing that, HN is a tetramer, has been identified crystallographically and on the surface of transfected cells as a tetramer. It's a dimer of dimers. And it exists either in a form where the dimer of dimers, and there's a small transmembrane region, very tiny cytoplasmic tail, a stalk, and then globular heads, and this is depicting the tetramer. And it can exist either in a form where the four heads are down in a state where they're partially contacting the stalk, or in a form where the four heads are up, freeing the stalk, and possibly in a sort of intermediate form with two heads up and down, at least been crystallized in that form. Fusion protein can exist in its prefusion state before it's been activated by HN to undergo the triggering, and then post-fusion after it has expanded and then refolded into its final stable state after the fusion event has occurred. And we have been very interested in asking, when HN engages receptor, where and when do HN and F interact? Do they physically interact? Is there a signaling event? And we've looked at interaction between HN and F in a number of ways. One of them have been using bimolecular fluorescence complementation, in which half of a fluorescent probe is attached to HN and the other half to F. And in this way, we can follow when and where the two proteins are actually interacting. And initially, it was thought that perhaps HN and F are not interacting before HN engages receptor, and then this brings F into interaction, thereby allowing it to be activated. Well, what we've seen, and these are photographs of bimolecular fluorescence complementation on transfected cells that are transfected with HN and F as a surrogate for the surface of the virus, assuming that what happens on the surface of infected or transfected cells looks like what happens on the surface of a virus. So that before receptor engagement, and this is a photograph in the absence of any receptor engagement, you can see that HN and F are interacting, based on the extensive diffuse fluorescence. When you allow receptor engagement to occur, however, those HNF interacting pairs cluster, patch, in the regions where fusion is going to occur between cells, because HN and F mediate fusion between a transfected or infected cell and an uninfected cell. So using this strategy and a number of others, we've shown that interaction between HN and F, the two surface glycoproteins, one with a receptor binding function and the other with a fusion function, they must interact before and then during receptor engagement, and that's a key regulator of fusion during entry. So to understand the stages of fusion activation, we developed some quantitative assays, and this one I'm going to show you a few experiments using. This is the same diagram that I showed you before, and at the first step, where nothing has happened yet, virus is not attached to the target cell at all. In the second step here, virus is attached to the target cell by HN alone, and then after F has been triggered by HN, virus is attached to the target cell by receptor binding, but also by insertion of the fusion peptide. And then in the last step, there's fusion. So if we use as a target cell, as a receptor-bearing cell, something that we can quantitate easily like a red blood cell, then we can measure very simply, quantitate each of these steps. So in the first step, we can quantitate how many red blood cells or receptor-bearing cells are not bound at all or released. We can then quantitate the percentage of red blood cells that are bound either by HN alone or by insertion of the fusion peptide. And we can quantitate how many red cells are fused. And so what we get is something, for example, over time, percentage of red cells that are either fused, released, the pool is going down and the bound pool is going down as the progression occurs through to fusion. And for a standard, what we call wild-type laboratory strain, by about 15 minutes, all the triggering and fusion has occurred. Now, we can then use this simple assay to ask which features of HN affect the fusion activation and modulate entry of virus into cells. Just as a reminder, we can identify HN with its heads down, covering the stalk, or HN with 4 heads up, this dimer of dimers with 4 heads up. And now looking down on the top of a dimer of globular heads, so looking down on the top of this, I'm showing you sialic acid, a sialic acid analog, nestled in what we have begun to call the receptor binding site 1, or the primary sialic acid binding site of HN. And this had been identified crystallographically, and also we've identified functionally that this is a primary receptor binding site. Now what we've also identified is that at the dimer interface between 2 HN monomer heads, there's a very important second binding site, which is, as I'm going to show you, involved not only in binding but also in activation of F. And many of these functions have been elucidated through the use of HN mutations, individual amino acid mutations in HN. Remember, F, the fusion protein, the one that's actually doing the fusion, is constant. But we're looking at individual mutations in HN that arise under selective pressure, which I can tell you about later, and that dramatically change HN's ability to activate F and initiate the fusion process. So for example, individual mutations that occur in either site 1 or site 2 of HN result in avid receptor binding and increased fusion. Individual amino acid changes that occur in site 2 that specifically increase HN's ability to activate F, independent from its ability to bind receptor, increase fusion. And a mutation in the stalk of HN, one single residue, decreased F triggering by HN and dramatically decreased fusion, showing us for the first time that the stalk of HN is actually critical for somehow inducing F to be activated by HN. So these gave us clues to HN's role in fusion promotion. For example, if you look again, this is the same graph I showed you. For wild-type HN, fusion is complete within 15 minutes. If you look at that HN with one single amino acid change in the stalk, it's incredibly much slowed down to the point where not till 45 minutes are 50% of the red cells fused in. There's decreased F triggering by HN and decreased fusion over time. Slightly different assay, a cell fusion reporter gene assay. We're looking at these two mutations, individual amino acid mutations at site 2, that dimer interface region of HN that cause increased fusion triggering, dramatically increased fusion over time compared to wild-type HN or that stock mutant HN that is highly handicapped. So we had these two mutations in site 2 that gave us these HNs based on these individual mutations that were just super triggering. We have viruses bearing HNs with these mutations that were just superfusers, very active in triggering. The viruses would just fuse through a monolayer. I don't know if anybody has ever done a plaque assay or infected a monolayer of immortalized cells in a flask with virus. You see individual plaques where the virus is fusing cells. These were just, you could practically watch them fusing through a monolayer. So what HN is doing, in order for F to be activated, you can activate F with temperature. You have to put in energy. HN is reducing the energy barrier for F to be activated, so that when we look at the same graph, except that now we have temperature on the x-axis, the temperature at which 50% of the red cells are fused in for wild type is 20. When we look at this HN with a single amino acid change in the site 2, the temperature goes down to 18, and that one, the superfuser, with another mutation, with one individual mutation in site 2, the temperature is now at which 50% of F has been activated or red blood cells have been fused, is now 15 degrees, down 5 degrees C. They are much more efficient at activating F and can do so at a lower temperature. And viral entry is also more efficient. Viruses bearing HNs with the single amino acid changes are able to enter cells at lower temperatures. They're more efficient at activating F. So these biological adaptations of virus entry process through selective pressure, evolution, and culture revealed many specific functions of the entry glycoproteins, revealed the fact that HN triggers F upon receptor engagement by the primary binding site, revealed that HN and F are interacting and that more or less strongly, but are interacting prior to receptor engagement, and revealed that a second receptor binding site is involved in activating F, and that if that receptor binding site is altered in certain ways, you can get just dramatically what we thought was better, more active fusion and entry. So we turn to asking whether or not these properties of HN, these features, are relevant in interplay with the natural host, because after all, the virus has to infect the lung. And so we took that, our original virus, before the selective pressures, and compared it with that superfuser HN site 2 mutant, and tried to grow these in airway. And that superfusing mutant was just dead, didn't grow in airway at all. One residue change in HN, everything else exactly the same. Superfuser in culture, didn't grow in airway, and didn't grow in cotton rat, which is a small animal model of respiratory viral infection for these viruses. So then we allowed selective pressure in the airway. So right here, these viruses are just barely growing at all, and we allowed selective pressure in the airway, and after a while, a new mutant.
What does that mean? How do you do it?
Let them grow. Let them try to grow in the airway. Let them just suffer through having to try to grow in an airway. And there they were, doing their best, and what happened? This was the temperature versus red blood cell fusion assay, looking at the temperature at which 50% of red blood cells have fused in, so 50% activation. Here was that superfuser able to fuse, able to activate F at lower temperatures. The first mutant that emerged during growth in the airway for adaptation, so sort of reverse adaptation to being able to grow in an airway, was so inefficient at triggering that 50% fusion occurred up here at 30-something degrees. And the final stable variant that emerged that was able to grow in an airway was so inefficient at fusing that even at 40 degrees, this guy wasn't fusing. Now some of you are going to notice this one that was able to grow in the airway that reverted itself to ideal growth in an airway seems to be even less fusogenic than our original. Yes, we'll get back to that. The first mutation that occurred during the efforts of this virus to adapt to growing in an airway was actually in the fusion protein and decreased the triggerability of the fusion protein. So it was actually a mutation that immediately protected F from being so readily activated. And the next mutation that occurred that created a stable, solid virus that was good for the airway was right in that site 2. Here was the original mutation that made it such a superfuser and unable to grow in the airway. And here, parental one. And right here, right next to it, in that dimer interface was a second mutation that made it able to grow in the airway and reduced its fusion. So the residues at the dimer interface, the airway disabled mutant, so the one that is superfusogenic in culture and couldn't grow in the airway, is able to more tightly interact at the dimer interface, whereas it appeared that or we hypothesized that when the emergence of this double, the second mutation, created a steric clash with a residue that was an arginine here that perhaps was pushing the dimer interface apart. And in fact, we then asked whether or not this affects dimerization, the second mutation that enables growth in the airway. And working with Ian Wilson and his postdoc at the time, Rui too, Rui looked at, this is diagrammed, the dimer interface, and the red are the residues that are interacting with monomer to monomer at the dimer interface. And in fact, with a doubly mutated HN, there are less residues interacting in the dimer interface even than the original wild type, and far more than with the original parental mutant. So knowing that the dimer interaction has been reduced in an effort to be able to grow in an airway. Sorry for anthropomorphizing the virus. It's not really making an effort. But in order to grow in the airway, we asked then, does this affect HNF interaction? And in fact, looking at the quantitation of the bimolecular fluorescence complementation assay I showed you before, with the superfuser variant and even the superfuser variant with the mutation, the transitional mutation in F that decreased its triggerability, HNF interaction is up here. Reduced, HNF interaction is dramatically reduced in the virus that is able to grow in the airway. And not only is HNF interaction reduced, but in vivo replication is permitted in the cotton rat. So the original strain, the superfuser in culture, and the revertant that can grow in the airway. So these site 2 residues on HN are modulating growth in vivo. Triggerability of F and activation of F by site 2 on HN clearly are essential. So did we just evolve a virus that was fit for humans by understanding the features of what it takes to grow, learning about what sites on HN and F are important? Did we end up evolving a virus fit for humans? So yes, we looked at clinical isolates of parainfluenza virus, circulating human strains, and analyzed these not only for sequence, but for their properties of fusion, fusion triggering, interaction between HN and F, receptor avidity, and also receptor cleavage, all properties that relate to the HNF interaction. And we found that they are exactly like the double mutant. The clinical viruses are less fusogenic, less avid, less able to trigger F. F is less triggerable. And in fact, there's a direct relationship between the reference strain that we're originally using, the airway adapted and clinical isolates. HN is the predominant determinant of whether or not these balance of properties are able to be fit for humans, and F also being less readily triggered contributes to that. So when you look at clinical strains thrown in there, there's an inverse relationship between fusogenicity, so ability to be a really good fuser, to bind, to enter, to spread. There's an inverse relationship between fusogenicity and ability to grow in vivo. And the best growth in vivo is the clinical isolate that we found. Next best are the ones that are adapted for growth in the airway. So if enhanced fusion activation is a detriment to survival in the actual host, we asked, so how is fusion triggering kept in check? The virus is encountering sialic acid-containing molecules all the time in the lung. There are mucins sialic acid-containing molecules. So what we found is that actually, so we knew that HN and F are together before receptor engagement, but that HN activates F only upon receptor engagement. We found that prior to receptor, yes, and also an assay that we use to determine when F has become activated based on acquisition of protease sensitivity, and yes, that there's another function, there's a fourth function of HN, this amazing molecule, which is that prior to receptor engagement, it stabilizes F. It retains it in its metastable state, preventing it from getting activated before the right time and place. As I mentioned, a lot of these experiments were done using transfected or infected cell surfaces as a surrogate for the surface of the virus. So we decided to look at what's going on the surface of the virus. And these experiments were done by, or these EMs were done by Kelly Lee, who you saw. Now, this is just in the negative stain tomography of a paraflu-3 virion. What we saw, what I'll show you, are that the surface glycoproteins are very heterogeneous, and that we see arrays of HN alone, which we don't know what they're for, and we see some patches of F alone, and then we see many arrays of HN and F together, as we did on the surface of the cells. Now, as you recall, HN can be present with its heads down or with its heads up, and there was a prevailing theory that was controversial, that perhaps HN and F, okay, they're together, but the heads of HN are down. And then upon receptor engagement, the heads of HN flip up, allowing the stalk, which we'd shown to be important for triggering, to contact F, and that's what happens. Well, what we saw on the surface of the virions is that HN alone is in patches with its heads down. The F and HN clusters, the HNs are all up. The heads are all up, and this is in the absence of receptor engagement. Sorry, I didn't mention that. So no receptor engagement going on at all. The HN heads are all up, and the F is all in its prefusion state. These are the patches of HN, and you can see these glorious patches of HN, all of them with their heads down. When we see HN and F together, you can see these arrays of F in its prefusion state with the globular heads of HN towering over them, and this is representation made by Kelly of the globular domains of HN towering over the prefusion state of F. And this is confirmed by radial density measurement of the radial density distribution, confirms that when you have patches of HN and F together, HN measures in its heads up distance from the surface of the virus and is present together with prefusion F, whereas when the HN arrays alone, HN is measuring in its heads down distance from the surface of the virus. So HN is not receptor engaged. HN is not signaling F. F is prefusion, but HN is in its heads up. So we think, and this is something I think that I'd love some help with from some of the people here, that HN, upon receptor engagement, the stalk requires a signal via site 2 that I showed you from the receptor engaged globular domain. And how this signal is occurring upon receptor engagement, since it's not from the heads flipping up, is something that we don't know, and we're very interested in learning. In the last few weeks, we're analyzing the results of a really fortuitous, great experiment, I think, in which a resident at University of Washington Hutchinson Cancer Center, the microbiology lab there gets lots of clinical samples of respiratory viruses, and they took 165 samples of parainfluenza viruses from patients. And Alex Greninger, who's a resident in laboratory medicine, directly sequenced, direct from the patients, 165 viruses. And those samples were split, and half of them went for direct sequencing, and the other half passed through standard microlab culture, where they culture the viruses to look at them for 2 days, and then they sequence them. So we've got samples that were directly sequenced and samples that went for 2 days through culture. Do you think that they back evolved? Do you think that they came from patients? Was 2 days enough for them to completely change their entry properties? And where do they change? Mutations in HN at the dimer interface, site 2, after only 2 days in culture. So mutations at that 552 site that we know from before. And we expected that. But then, those are from right here. On the other side of the dimer interface, there's a whole new set of mutations in site 2 that occurred just in those 2 days of culturing of those clinical samples. And we're looking forward to understanding the functions of those site 2 dimer interface sites, and we're hoping they will clue us into how actually HN is activating F. So we think that in addition to studying the details of the structures and functions in systems that we can control, it's important to look in natural tissues and study authentic entry mechanisms in lung tissues. And so one model that we've been using is a human airway epithelium, which grows at an air-liquid interface. And these are adult tissues. They're large airways and bronchioles, and we've derived a lot of information. That's what I showed you where we realized that the superfusers weren't growing in airway. This is what we used. But now, in collaboration with Hans Schnuck, who's a stem cell biologist at Columbia, we're developing a 3D model. These are human lungs differentiated from pluripotent stem cells. And what we get are lung bud organoids that recapitulate, in many respects, lung development. They develop small airways and distal alveolar structures. And they are compatible. They have branching morphogenesis, and they develop up to late second trimester, so about 24, 25, 26-week infant. And then they seem to stop their maturation. They're mostly distal lung, and they have alveolar type 2 cells. So we really think that they are going to provide us with a model of premature infant lungs. And premature and young infants are who are predominantly get the worst effects of these viruses. So we think this is going to be a useful model. Proof of concept, we were infected with respiratory syncytial virus, this fluorescent virus infecting these airways and being shed into the lumen. And then we showed, looking at individual cells infected with respiratory syncytial virus, which is another similar pathogen, that the cells are infected and then shed into the lumen of the airway, which is exactly what happens actually in infected humans. We're now developing this into an authentic representation of paraflu infection. We were micro-injecting virus into the lumen of these lung bud organoids to try to represent infection of the apical surface. It has a ways to go, but we're excited about this. We've been able to show the infection and spread of fluorescent live virus in a pattern that we think is similar to what happens in infants, although not much is known about that. So we're hoping to really change what's known about infant infection with these viruses. Understanding principles that are important for infection in a natural host, we think is going to guide ways of interfering with natural infection. So now that we know that overactive triggering is a handicap, the superfusers were really handicapped, we understand that we could use perhaps a receptor analog to prematurely induce HN to trigger F, activate F, and thereby inactivate virus before it could get to its target cell. And we've developed some proof of concept molecules, including this one, that is able to induce HN to prematurely activate F. So here, as I sort of mentioned in response to Bill's question, when F is activated from its metastable prefusion form to its transient intermediate, it becomes protease sensitive. So when HN is receptor engaged and it activates F, F is protease sensitive. When HN is protecting F, then when there's no receptor engagement, F is protected and is not degraded by protease. It's holding it stable. But even when there's no receptor engagement, so HN is there presumably protecting it, if we add this small compound, F transitions to protease sensitivity, so it has become triggered. So the small compound is inducing HN to activate F. And in fact, in airway epithelium, the small compound reduces the titer of virus, and the virus is inactivated. So this is just a sort of a proof of concept so far, but we think it could potentially be an exciting strategy that might work in the natural host where the receptor analog leads to premature activation and inactivation of virus. So we think that strategies also we're confident that strategies that might be used for inactivation of viruses will need to be assessed in natural tissues as well. So HN and F, as I hope I've showed you, are in this incredible balance of properties where only the right balance of properties actually end up in a virus that is able to infect its natural host. Receptor avidity, if it's overly avid, as in some of the superfusers, results in excessive triggering. HNs that are super triggering look great in a dish. F that's more triggerable looks great in a dish. If F stabilizing feature, so the feature of HN that holds F in its metastable state prior to receptor engagement, is decreased, you get too much activation of F. And if receptor cleavage mediated by HN, something that we haven't really talked about much, but if HN's decreased in receptor cleavage, then there are too many receptors around, too much left for interacting with HN and activating F, and that makes a virus that's too activatable also. All these features are in variants that have evolved in culture or that have evolved in other tissues, for example. Receptor cleavage is very low. In clinical strains that you find in the field, they have very high receptor cleavage function because they need to not have a lot of receptor available to constantly be activating F. So only when all these features are in a really precise balance that's just suited for the host do we get infection of the lung. A lot of people have contributed to our work in general and to the work some of the work that I described here. This is my group at Columbia, and these two people are the ones that are developing the stem cell model of lung. Kelly Lee worked with us on the cryo-EM and the EM of viral surfaces, and Ian Wilson on the structure of the dimer interface. Alex Greninger is working with us on the comparison between clinical and lab-adapted strains that have been cultured in the microlab, and all the evaluation of the differences that's showing us about what it takes to be a real virus. Thank you.